Hello everyone, this video is here to explain CRISPR-Cas9 based targeted genome editing. This video will cover following topics. Basic introduction about CRISPR-Cas9. Evolution of CRISPR as genome editing tool. How this system works. Designing of guide RNA. Applications of this technique in the life science field. Before the discovery of the CRISPR-Cas9 system, artificial fusion proteins zinc finger nucleases, ZFNs, and transcription activator-like effector nucleases, TALENs, were the tools of choice for targeted genome editing. Both ZFNs and TALENs are essentially bi-domain nucleases comprising of a target-specific DNA binding domain responsible for target specificity, fused to the fork-1 restriction enzyme nuclease domain cleavage action. The introduction of CRISPR-Cas9 system is replacing the pre-existing site-specific nucleases, ZFNs, TALENs and meganucleases, and becoming a preferred genome editing tool for most of the species. So before understanding about targeted editing mediated by this new strategies called CRISPR-Cas9 system. Basic idea about discovery of CRISPR-Cas9 system. CRISPR is clustered regularly interspaced short palindromic repeat. CRISPR-Cas systems are composed of Cas genes and CRISPR array consisting of genome targeting sequences called spaces interspersed with identical repeats. CRISPR-Cas9 system derived from bacterial defense system that protect organisms from foreign DNA. When a foreign DNA or virus such as bacteriophage infects bacteria, bacteria integrate a stretch of that foreign DNA into its genome between two adjacent repeats, located at the proximal end of the CRISPR locus. Following subsequent encounters with foreign DNA, small interfering CRISPR RNAs called crossing RNAs are processed by transcription of spacer comprising CRISPR arrays. CRRNAs combine with trans-activating CRISPR RNA called CRRNA to form guide RNA complex which activates and guides Cas9 nuclease to initiate the double-stranded break in the foreign invading DNA known as protospacer. Scientists utilized this bacterial defense system for targeted editing. They engineered the CRISPR RNA to be complementary to the target sequence in the gene. And then the programmed CRISPR RNA make complex with Cas9 and bind to the target site by complementary base pairing and hence cleave the target site by inducing double-stranded break. This double-stranded DNA break will be repaired by a non-homologous end joining pathway, which is an error-prone repair pathway. As a result of which insertion or deletion mutations occur at the target site. It has two main components. One is guide RNA, another is Cas9 endonuclease. We will discuss about each component separately. The Cas9 system require dual RNA for target DNA binding and cleavage. One is the CRRNA and another is trans-activating CRRNA. Mature CRRNA base pairs to trans-activating CRRNA forms a stem loop-like structure that directs the CRISPR-associated protein which is Cas9 to introduce double-stranded breaks in target DNA. Trans-activating CRRNA might be required for target DNA binding or to stimulate the nuclease activity of Cas9 downstream of target recognition. Trans-activating CRRNA is required for target DNA recognition, possibly by properly orienting the crossing RNA for interaction with the complementary strand of target DNA. The predicted CRRNA and CRRNA secondary structure includes base pairing between the 22 nucleotides at the 3' terminus of the CRRNA and a segment near the 5' end of the mature trans-activating CRRNA. This interaction creates a structure in which the 5' terminal 20 nucleotides of the CRRNA, which vary in sequence in different CRRNAs, are available for target DNA binding. The bulk of the trans-activating CRRNA downstream of the CRRNA base pairing region is free to form additional RNA structure and to interact with Cas9. Cas9 is a DNA endonuclease guided by two RNAs. Cas9 proteins constitute a family of enzymes that require a base paired structure formed between trans-activating CRRNA and the targeting CRRNA to cleave target by inducing double-stranded break in the DNA. Cas9 recognizes a specific sequence called protospacer adjacent motif or PAM and induce a double-stranded break 3 base pairs away from PAM. Cas9 uses two nuclease domains to cleave the two strands in the target DNA. Cas9 contains domains homologous to both HNH and RUVC endonucleases. Cas9 HNH domain cleaves the complementary DNA strand, whereas the Cas9 RUVC-like domain cleaves the non-complementary DNA strand. As we saw in bacterial system, CRISPR defense system involved two RNAs which are CRISPR RNA and trans-activating CRISPR RNA. These two RNA molecules which hybridize to ensure Cas9 activity at the target site specified by the CRISPR RNA. They were merged into one molecule called single guide RNA to facilitate cloning. Hairpin structures within the single guide RNA mimics the CRISPR RNA and transactivating CRISPR RNA complex enabling the binding of Cas9. A guide RNA is an engineered RNA molecule containing two elements, a CRISPR RNA which contains the target site of the genome and is usually 15 to 20 BP long, a trans-activating CRISPR RNA, which binds to the Cas9 nuclease Cas9 endonuclease programmed with single RNA molecules to cleave specific DNA sites, provide a simple and versatile RNA-directed system to generate double-stranded DNA breaks for genome targeting and editing. A typical CRISPR-Cas9 workflow involves many steps. They can be divided into the following steps: design your guide RNA and select a Cas nuclease. Cas9 nuclease can be an ideal choice for the generation of knockouts. Then cloning of guide RNA. Then introduction of CRISPR components into the system. Then appropriate selection and screening of candidate genome edits. Lastly, verification of your genome edit. So first step is selection of target gene. Target gene can be selected from published literature, in data from omics and reverse genetics studies. Guide RNA designing. Guide RNA selected should be target specific and have no or very few off targets. A wide range of software are available both online and offline, predicting sequence, efficiency and even off targets for the input sequence. Commonly used software includes Cas Analyzer, ChopChop, and CRISPR-P. Delivery of guide RNA and Cas9 by transformation of cells. This step involves the introduction of Cas9 and components into the cells. The concentration of Cas9 nuclease and guide RNA needs to be optimized to provide the highest possible mutagenesis frequency. Screening of mutants. For editing the mutants can be screened using Sanger sequencing, or next generation sequencing. In the next video we will discuss about each step in detail. And learn designing of guide RNA. Softwares for designing guide RNA. Selection of Cas nuclease. Modes of delivery of Cas9 and guide RNA. Applications of CRISPR-Cas9 system. Limitations of CRISPR-Cas9 system. I hope you like this video. Please subscribe to this channel and like this video.